In chapter 15, we're going to talk about several parts of the chapter that really relate to understanding that the inheritance patterns that we see in organisms really come from the way that chromosomes behave during gamete formation. So if we go ahead and get started, right, when we talked about Mendelian genetics in chapter 14. Remember I prefaced that when Mendel was doing his crosses and when he was coming up with his model, he really had no idea that chromosomes, genes existed. And so he coined this term hereditary factors. And we now know that these hereditary factors, while they were purely abstract to him, we now understand that they are genes that are located on chromosomes that get sorted during gamete formation. And we can sequence genes now. We can visualize condensed chromosomes when you do a karyotype, for example. We can also use fluorescent markers to actually pinpoint exactly where on a chromosome a gene is located. And that's kind of what you see on the image on this slide. So we've got some condensed chromosomes that have been taken out of a cell. They've been stained with a red-orange fluorescent dye. And then we have made a fluorescent probe that can specifically find a certain gene on one of these chromosomes. And that fluorescent probe, here you can see it's shining in yellow. And this particular gene exists right here in that position on this chromosome. Now you'll notice there are two yellow spots, right? This is because this chromosome is a duplicated chromosome. It's been taken from a cell that's actively going through mitosis, which means it's gone through S phase and it's condensed all of its chromosomes in preparation for going through that complex process of mitosis that we talked about in chapter 12. All right, now one of the pioneers in genetics that really helped us solidify this idea that certain genes are located on certain chromosomes and the movement of those chromosomes during gamete formation is important when we look at heredity and how traits are passed on from one generation to the next, was this guy by the name of Thomas Hunt Morgan. He was one of many scientists, and if genetics really piques your interest, I encourage you to read all of chapter 15. There's a lot of really interesting information here, a lot of really interesting fruit fly genetics experiments that were performed that we're not necessarily going to go through for the sake of time, but it's all in there. So, if this is something that, is your bag of chips, by all means, read those sections in chapter 15 that I don't assign to you necessarily. All right, so let's take a look at one of the experiments that Thomas Hunt Morgan did. Now, Morgan was maybe not as lucky as Mendel was in terms of his choice of model organism, right? Mendel decided to work with peas. They were easily accessible. He could find different varieties to cross. For Thomas Hunt Morgan, it was a little bit more difficult. You see, the fruit fly that he chose to work with, which was honestly a great genetic model because they breed like crazy, produce a lot of offspring. Their genomes are really simple. They only have about 8 chromosomes in there. And they're even to this day used as a good genetic model organism because their genome is so easy to manipulate. So all that was great. The trouble for Morgan was that all the fruit flies he had were identical in terms of their phenotypes. They all looked the same. So he didn't have different varieties that he could cross to each other to see what the outcome for the offspring would be. So honest truth, he spent like the first 2 years of this project simply just breeding fruit flies so that he could get some sort of mutant to appear naturally, spontaneously, so that he would have different variety flies to cross. Now, I don't know about you, but I would not have lasted 2 years breeding fruit flies. So I really give props to him for sticking with it and getting his mutants. And his mutants were flies that had, instead of red eyes, they had white eyes. And so this experiment shows you kind of what Morgan did. He simulated what Mendel had done in terms of the crosses. It's a very, very similar experiment. And we have it summarized here on the left-hand side. So he went through 2 crosses. He had his P generation flies, right, his true breeding variety flies. So he took a red-eyed female and crossed it to a white-eyed male. And in the F1 generation, he noticed that all of his flies came out with red eyes, indicating, complete dominance, and showing exactly what Mendel had found with, for example, his flower color pea plant crosses, right? He crossed a purple flower to a white flower, and all the F1 offspring had those purple flowers, much like all these flies had red eyes. He didn't stop there, being a good scientist, and he crossed the F1 generation to themselves or to each other, males and females, and then he got the F2 generation. And here, again, just like Mendel, he saw a 3-to-1 ratio of red-eyed flies to white-eyed flies. Except there was a little bit of a hiccup here. While he had the 3-to-1 ratio, he also noticed that the white-eyed flies that he got were all males. It's kind of weird, right? You would expect that, 50-50, you'd have 50% of the red-eyed flies females, 50% would be males, and the same thing would go for the white-eyed flies. But no, he couldn't get a white-eyed female from this particular cross. He's like, well, that's really curious. I've got to figure out why this is. And so Morgan did. And his explanation was that the gene that codes for fly eye color is actually located on the X chromosome in flies. So you may or may not know that gender is determined in flies just like in humans. So we've got an X and a Y chromosome, so do the flies. If a fly gets 2 X chromosomes, it's a female. If it gets an X and a Y, it's going to be a male. And so Morgan determined that this eye color gene happened to be on the X chromosome. And he was able to use this idea to explain exactly how he got the results that he got in that F2 generation result. And so that's kind of outlined here on the right-hand side. So let's just go through it briefly. So here in the P generation, right, he took his red-eyed female, so 2 X chromosomes, and each chromosome, because this is a true breeder, both X chromosomes would have the red-eyed allele. And he crossed that female to a white-eyed male. Male gets an X and a Y chromosome. Y chromosome doesn't carry the eye color gene, and so in this case, if this is a wide-eyed fly, then the only X chromosome that the male has needs to have that wide-eyed allele. All right, so now the female and the male would make their gametes. All of the gametes in the female only has the red-eyed allele to give, so all of her eggs would have an X chromosome with that one dominant red-eyed allele. For the male, 50% of the sperm will get the X chromosome, 50% will get the Y chromosome, and you see that here. When you do your crosses, here you have your females having the 2 X chromosomes, and these would be red-eyed because they have that one dominant red-eyed allele. When you do this cross here, you get the Y chromosome from the dad, the X chromosome from the mom, and all of these male flies now also are going to be red-eyed because they get the red-eyed allele on their X chromosome from the female. So far, so good. Now here's where we get a chance to take these F1 generation flies and cross them to each other. So when the female makes her gametes, 50% of the eggs get the X chromosome with the red-eyed allele, and 50% get the chromosome with the white-eyed allele. For the male, when the male fly forms the sperm, 50% of the sperm will get the X chromosome with the red-eyed allele, and 50% of the sperm will get the Y chromosome with no eye allele. And so now when you do your 4-square Punnett square here, you'll notice that all 3 of these flies are going to be red-eyed, right? You're going to have these 2 are going to be females. One is a homozygous dominant, one is a heterozygote, and then this is going to be a red-eyed male. And the only way that you could get a white-eyed fly is to get one Y chromosome from the sperm and then inherit that one X chromosome with a white-eyed allele to make a male fly with white eyes. All right, so Morgan determined that the white-eyed mutant allele must be located on the X chromosome based on the inheritance pattern he saw. He was one of the first individuals that actually, got a chance to say, hey, this particular gene is located on this chromosome, and the inheritance pattern helps to support that idea. All right, now this particular slide is a really good slide for review. It looks really complicated, but it's talking about a lot of the information that we covered already in chapter 14. So looking at the law of segregation and the law of independent assortment as Mendel defined them. And this is all using Mendel's dihybrid cross. So looking at his cross between the yellow and round and the green and wrinkly parent generation peas. I actually have a separate video for you guys to check out that goes through this exact same process, except instead of using Mendel's peas, we're using the Black Lab exercise or example that was presented to you in chapter 14. So I encourage you guys maybe to even pause the video here and go check out that Laws of Inheritance extra video for chapter 15 to help review this particular slide and the two laws of inheritance.